Good morning, everyone. Welcome to this workshop on COVID-19. This is going to be a workshop on the genomics side of the novel coronavirus. This workshop is designed for teachers, students, and their parents. We hope that as a result of this workshop, you also can get excited about bioinformatics as a good way to study biology, not having access to a lab or being able to conduct experiments, especially not with this new virus. In this workshop, we will speak about the genomic data analysis, and we'll try to understand how different data analysis tools can help us detect viral genomes in next-generation sequencing data, help distinguish between different viral strains. For example, how is this novel coronavirus different from other coronaviruses? How we can identify specific functionally significant changes in the viral genomes, and in general, how can we discuss virology from a data perspective? So some of these elements are listed here on the screen. What you see here is the coronavirus and the message that it carries in its RNA. We can see the code of this message in the nucleotide sequence. We can also study relationships between different coronaviruses, which is what you see right here in the back, the phylogenetic tree that we'll talk about. And then how do we understand the biological significance of the different variations that we actually find? And in the middle, you see some of the tools that we'll be talking about. And some of those tools include multiple sequence alignment, as well as phylogeny, how to study relationships of these different viral sequences. Before we start, though, I want to mention that this workshop is organized as a collaboration between Pine Biotech and ScienceCoach. And so I want to give a few minutes to Jill, who is the founder and executive director of ScienceCoach, to introduce her organization and share with you more of the details about the resources and programs that are available for teacher training and high-level student research projects. Jill, I'm going to unmute you now.
Good morning, everyone. Like Elia said, my name is Jill Malcolm. I'm the Executive Director of the ScienceCoach program. We are a program that allows 6th through 12th grade teachers and students to really explore some type of science that they are particularly interested in and to do research that allows them to take that research to another whole level. So we allow the students to choose a question of personal interest. They create a procedure to test that hypothesis or to develop that concept, and then they arrive at answers with a whole new level of validity, and it really builds confidence in the students to help them learn how to solve real-world problems. We work with students 6th through 12th grade in public, private, and home schools in the United States at this immediate moment. So a year ago, we started a pilot with Pine Bio to try to bring the whole science of bioinformatics to high school students. We have a informatics website, and that link is in the chat. And you're welcome to use the courses that we have up there for free. The results that we had were absolutely astounding. We had students who knew absolutely nothing about bioinformatics, 2 and 3 months being able to run their own data through using the Pine Bio system. That's nothing short of miraculous. These students, we actually had one, Hannah, that did a gene expression experiment in diabetes and looking at actually identifying not only that gene, but figuring out how to allow pharmaceuticals in the future to be able to suppress that gene expression, which in layman's terms could actually provide the key to allow gene-based diseases like diabetes to be cured. So this is real research that the students are doing, and in order to support the students, we also train the teachers. So we have a summer training program going on. This June and July, we will be doing some virtual training, and we invite anybody who's interested in being a part of the ScienceCoach program, anybody who'd like to do high-level research with their students, and then those projects are entered into competitions. And then sometimes if they come up with a novel idea or a patentable idea or an idea that can turn into a business, we actually help them do that. So the relationship with Pine Bio has been phenomenal, and we continue to increase the availability for this content and making it adapted to a 6th through 12th grade student.
All right, great. Thank you very much, Jill. So if anyone wants to learn more about ScienceCoach Bioinformatics, there's a link that points to this website. This website has some materials that you can start looking at, but I also want to mention that ScienceCoach is beyond just bioinformatics, and you can look them up on Google, or I'm sure that Jill can post a link to the ScienceCoach website. Okay, so let's start talking about COVID-19 and what we will talk about in this session. So COVID-19, the name stands for coronavirus disease, and 19, of course, is the year that this epidemic started, 2019. So what we'll do in this workshop is we'll try and look at some of these phenotypes, some of these things that characterize this virus. So what is this coronavirus itself? How does it cause the disease? And what is the result of viral transmission from animal reservoirs where potentially that virus came from? And how does that enter into the human cell? And we'll talk about all of these things from a perspective of the collected data that has been publicly available since 2019 for this specific virus, the novel coronavirus, but also for a long time, for the past decade, available for other coronaviruses. And so we have, as a result of this publicly available data, any one of us has access to very accurate sequences of the genomes of these viruses produced by top virologists and labs around the world from many different locations, including China, the US, Italy, Israel, Africa, and other places. So we really have access to a unique repository of publicly available data, and we are the public, so of course we are able to access this data. But the question is, what do we do with this data, right? So the question is, how do we actually make use of the data? How do we analyze these genomes? And what is it that we can understand from such analysis? So these types of programs are supported by a number of different universities in the US and around the world that Pine Biotech is helping develop materials and tools to support their research and support the learning activities that they have for bioinformatics. I'm sorry, somebody is here annotating my screen, so I'm going to try and pause that. Beepsha and Mohit, maybe you guys can take a look at how to erase those. I think it's somewhere here in the okay, here, I'm going to clear all drawings. All right, great, thank you. Okay, so as I was mentioning, Pine Biotech is developing a number of resources that include some tools as well as curriculum that people can, that different universities use to teach and support research projects. And these include an online web-based platform that could be used for different types of tools, different types of analyses, for example, multiple sequence alignment and phylogenetic analysis that we'll talk about today. But also we offer a variety of different courses that span different types of data sets For example, next generation sequencing data that we'll talk about today in the context of genomic, transcriptomic, metagenomic studies, but also provides tools for analysis of other types of data, including structural data analysis and mass spec and others, providing big data capabilities and machine learning capabilities to novel users that would like to use tools without having the technical engineering background, but also providing high-level research tools for big data for researchers that study a variety of different projects. So what we'll cover in today's session are several topics that are extracted from a full program that we have on bioinformatics for infectious diseases, where we take projects from published data sets and we reanalyze that data so that we can understand how to analyze, how to interpret the analysis results, and how can we use bioinformatics to study various aspects of viral infections of different pathogens. So this full program is going to start on May 4th. I would say that it's more at a college level program. However, what we've seen are a lot of students, as Jill mentioned, when they're introduced to some of these ideas and these tools, they're absolutely capable of taking advantage of these tools and making them, turning their own projects into something that is competitive. So the full program, again, will start on May 4th. Make sure to check that out by following that link that Beepsha posted in chat. Okay, so in today's session, we will talk about zoonotic spillover, or how different viruses can originate in animals and transfer to humans. We'll talk about the cell entry and evasion of the immune response. How do the viruses do that? We'll talk about how they produce proteins and replicate using the host cellular machinery. We'll talk about different analysis approaches that help us detect viral genomes and next generation sequencing data. When you take a sample from a bat, a pangolin, or a human, how do you know that there is a virus using next generation sequencing? We'll talk about viral strains and haplotypes. So we often talk about FASTA files of those assembled genomes. And in those genomes, we really cannot find a lot of the detailed variation that typically occurs in patient samples. We'll also talk about how to find, annotate, and characterize different mutations. So what are mutations, different types of mutations, and what is their significance based on the change that they produce? We'll also talk about how to find and use some of the data sets that are available online. So we'll actually go to a database where we will find some sequences from the SARS, MERS, and other coronaviruses that are available. And we'll talk about how to specifically find the COVID-19 samples. And then we'll also look at different hosts, animals, humans, different types of animals, etc. Okay. So the coronavirus has been in the news. We all heard and read a lot of publications and news stories. We probably saw a lot of statistics, but we will be talking today about the coronavirus from a genomic perspective, right? So I want to kind of make sure that everybody is focused on looking at the coronavirus, not as a particle that necessarily causes disease, but first at the basic level. What is the message? What is the message that the viral particle carries? So we already started talking about this. There is a sequence, and this is a sequence of nucleotides that is tightly packaged in something that is probably similar to a droplet of oil in water. So imagine a droplet of oil, and inside that droplet, there is a message. And that message can be studied. So the sequence of that message can be studied by looking at each individual nucleotide. Now we can also look at how that nucleotide sequence can be translated into amino acid sequence and then built into proteins. Of course, that does not happen by itself. To build those proteins, the virus has to access the cell of the host to utilize different elements of that cell machinery to build that protein. But we can look at different types of viruses. For example, they come from different hosts, And that means that their sequence should be different. If they're able to access the machinery from animals or from humans, they need to make adaptations. So they actually need to change that sequence. They really cannot change anything else. And that is the process of mutations, accumulation of those mutations, and the study of those mutations really are possible by multiple sequence alignment. And as a result, we can look at how those sequences are related to each other. And by that way, we can study how exactly are they different from each other or similar to each other, and build some kind of a sense of how are they interrelated. So let's talk about that relationship. Viruses are organized into groups by what's called taxonomy. So we talk about a virus like a coronavirus, for example. That really means many different things. In fact, coronaviruses have different genera, and there's alpha, beta, gamma, and delta coronaviruses, and they can be very different. In fact, some of them can only infect specific non-human hosts, and some of them are specific to humans. So there are different types of viruses. They have different properties. But at a deeper level, they are also divided into subgroups, and those are called species. And then within those species, there are additional variations. So in fact, a lot of these human coronaviruses, including types 229E, NO63, OC43, and HKU1, usually cause mild to moderate upper respiratory tract illnesses like the common cold. So most people actually get infected with one or even more of these viruses at some point in their lives. A lot of these viruses come from alpha and beta generas. And so when we say, I got a cold, that cold could be caused by multiple different pathogens. A good chance that if you had a cold, it was caused by one of the beta or alpha coronaviruses. So what about this novel coronavirus, right? This novel coronavirus is actually a specific strain. And that strain is a human SARS-CoV-2, coronavirus 2, and that belongs to the B species of the beta coronaviruses of the coronavirus family. So it is a very specific strain, and we now know from multiple genomes that have been made publicly available and already analyzed that even within the strain, there are now multiple strains that could be detected and specifically defined by their mutations, by the changes in their genome. So the differences in phenotype, the differences in how they cause disease are significant. Before 2002, it was thought that coronaviruses cause only self-limiting and mild infection of the respiratory tract in human beings. Two of the coronaviruses considered during this time were beta coronaviruses, and the other were alpha coronaviruses. They are endemic, so that means that they typically spread in a certain geographic area and cause about 15 to 30% of the infections of the respiratory tract each year in the areas that they are endemic to. So what that means is that typically a virus infects people in a restricted geographic area, and that causes mild symptoms. And so most people don't get tested, right? So we don't really know how widespread really the virus is until it starts causing severe symptoms. So for example, a previous outbreak of the severe acute respiratory syndrome, which is called SARS, happened in China in 2002 to 2003. And it was caused by the SARS coronavirus, which is also a beta coronavirus. At the time, this outbreak was among the most severe illnesses that had occurred due to a coronavirus. More than 8,000 people had that illness. During the epidemic, researchers obtained viruses that were closely related to the SARS coronaviruses from various exotic animals, such as raccoon dogs and the Himalayan palm civets. But it's generally accepted that the SARS coronavirus was first found in bats, right? So think about where this virus comes from, think about what it causes, the symptom that it causes, how it spreads, and how could that be an insight, a key for us to be able to study these different viruses. So in this session, what we want to do is really start talking about some of these concepts in order for all of us to start asking questions, right? So the goal of this webinar is not to give you a lecture. The goal of this webinar is for you to formulate your question, to know, for example, what questions can you ask? So not all questions could be asked from all data sets, right? We also have data sets that can answer very specific questions. So when you think of some of the topics that we discussed up until now, for example, where it came from, that is a question that we can ask from a very specific data set, and we can collect that data set, right? So we need to know how to coordinate the questions that we can ask with the data sets that will answer that question. But to answer the question, we need to have appropriate tools. So we cannot just ask a question and it will tell us. We need to analyze the data, and the data typically contains a lot of noise being a lot of information that does not answer the question that we're asking, and signal. And the signal are actually features, characteristics that do answer the question that we have asked. And then we will try to interpret the results, right? So after the analysis is performed, we will try to use additional information that could be found somewhere that will help us interpret the results that we got. Okay, so as I'm going through the next question, please try to formulate your own question, and then I will pause and I will ask you, and you will be able to share with everyone in chat what is a good question that you think that might be asked from the data sets that we will discuss today. So question number one, what is the origin of the novel coronavirus? So this novel coronavirus, we talked about that possibly it could come from an animal source, but a question would be which animal, what location, and how did it actually get into humans? So if we know something about bats, we know for sure that they have different proteins on the surface of their cells that are different from the human cells. And those proteins are essential for the virus to enter the host. So if there was a big change, how could that change happen? The question is more specific though, because if a change is big, then it's less likely. For example, if a whole section of the genome has to be completely different, that is not likely to occur. How do we know that? Well, the changes in genome occur by different processes, and it's important to understand these processes to be able to analyze the data. One of the processes that is very common are mutations. Mutations are single point changes, single nucleotide changes that could be either one nucleotide replaced with another one, and sometimes added or subtracted, inserted or deleted. And so those occur fairly often, and we actually know approximately what is the rate of mutations in positive strand RNA viruses. However, bigger chunks are typically not just gonna get replaced or changed in a specific strain. So how can a change like that happen? If it is indeed a bigger change, could it happen from recombination? Recombination means that a virus takes some portion from a different virus that is maybe in close proximity to where this virus is. So potentially we're talking about multiple strains of a coronavirus in a single host. And then we need to ask a question, well, could that really happen? So let's take a look at this genome. This is the genome on NCBI. So we can take this genome and find it on NCBI. So let's just take this and we will go to NCBI and take a look at this genome. I'm going to paste this link in chat. Oh, sorry. Okay, so please look at this link together with me, and let's take a look at what we can understand about this type of data. So what is NCBI? NCBI is the National Center for Biotechnology Information. It is a database hosted by the federal government that helps annotate all of the data that is published in different publications. So you will find here genomes, you will find here protein sequences, you will find here a lot of different information about everything that's going on. So here you can see right on top that a specific portion of this database called the nucleotide database is where we find this COVID-19 or SARS-2 coronavirus isolate, and it has an accession number, which is this code that you will find as we look at these different genomes. And then you can see some of the data about the sequence. So for example, you can see how long it is, 29,000, almost 30,000 nucleotides or base pairs. It's a single strand RNA. And you can see what is the date when it was last updated, which is March 18th. So now we can go here, for example, this is version 3 of the same genome, so it was submitted multiple times. You can see the taxonomy, where it belongs to, we talked about that. You can see who it was published by, and the publication where you can read more about the submission of this genome. And then you can see how it was submitted by a specific organization, and it actually replaced a different previous version, and so forth. And so then what's important about this is that you will see that a genome has a sequence, and that sequence has a structure, right? So the first question I'm going to ask you, what is this sequence? So as you look at these letters, let's try to use this to evaluate how familiar you are with genomic sequences. What are these letters? Can anyone tell me? Okay, great. Protein sequence, amino acids. Okay? Amino acids, great. All right, so a lot of people here are already familiar with how this works. So we'll talk a little bit more about that. And so what you indeed see here is that this is translation, right? So the process of translation is when we take a sequence from DNA, or typically a nucleotide sequence, could be from RNA, could then be transcribed. So DNA is typically transcribed to RNA, and then from RNA it's translated to proteins. And so we see here the translated version of a specific proteins. So you can see here CDS, which is the coding sequence for different genes, and they encode for proteins. So as you scroll down, you will see that there are different proteins, and down below, what is this? Who can tell me what this is? DNA, nucleotides, okay, great. So, okay, so these are sequences in nucleotides, that's what we want to get at. So let's take a look real quick, right? We said that this is about 30,000 nucleotide or base pair long sequence, right? So let's take a look at how many we can find right here. 29881, which is right here, and then it goes all the way right here, right? So this is, but we said that this is an RNA virus. So how could this be a DNA sequence if we're talking about an RNA virus? Reverse transcription. Okay. Anybody else? cDNA, great. ddNA. cDNA. Okay, so now let's clarify that. So how did we get from an RNA virus to C nucleotide? So the only change between RNA and DNA is uracil. And so what we typically record sequences of genomes is in cDNA format, because indeed the process of sequencing uses conversion to DNA to actually do to record those sequences. So that's why we see it like this. And the process of going from this sequence to protein is something that we will talk about as well. Okay, so great. So we saw the genome. So let's talk a little bit about how we got here, right? How did we get this kind of a data set? Okay. The approach that we will take to answer the first question. So let me remind you, what is the question? The question is, what is the origin of this novel coronavirus? So if you look at this submission, this was the first genome submitted by a group out of China of a novel SARS-2 coronavirus. And they submitted this whole sequence, right? And the question was, where did this sequence come from? So this sequence could have come, and here you see a couple of links. We will share this publication with you after the webinar. You can see that the question relates to how did it get there? What is the process of this sequence appearing in humans, right? So all of a sudden they found this and it appeared in humans. And the question is, okay, how did it get there? So potential hypothesis, right? Somebody formulated a hypothesis and said, okay, so maybe there was a primary host that originated in something like a bat. That bat maybe was eaten. Maybe there was an intermediate host, and eventually it got into humans, right? So here's a lot of questions that we can ask. Where did it come from? How did it get there? What was the process of getting there, right? So the only thing that we have to answer this question is really the sequence. We don't have a private investigator in China that can track the events back and really tell us how did it happen. All we have to answer this question is the sequence. But we're not going to do this on our own because we're not virologists. Maybe some of you are. I'm not a virologist. So we will follow an expert, somebody that has done it for us, and just to make sure that we're doing the right steps. After we do the right steps, we'll be able to think about it ourselves and ask additional questions. So here's a publication that was recently published in Nature Medicine, where they took several different sequences. So those sequences included human SARS-CoV-2, the sequence that we just looked at, included a couple of sequences from bats. So you can see bat, 3 of them right here. Another human SARS, so not the novel SARS, but the older one from 2003 to 2007, and a sequence from pangolin. And then by comparing these different sequences, they found specific changes. So what are the changes that we will see here? Those are 2 different types of changes. Some of the changes are single nucleotide or single amino acid, actually. And some of those changes are actually whole sections that were inserted, right? So that points to some biological mechanisms that can explain how this virus got from original host, which we are hypothesizing could be either bat or pangolin, into the human host. So we're going to follow this example and ask some of the similar questions. Okay, so, but for us to really understand how to answer some of these questions, we need to lay down some terminology. And some of that terminology has to do with how the virus enters a cell, replicates inside the cell. What is the mechanism of how that works, which is on the biology side. And some of it has to do with informatics or data science. So for example, we need to understand file formats. What is a FASTA? What is a FASTQ file? What is a PDB file? What are nucleotides? What are amino acids? And how can they be described when we talk about insertions, deletions, substitutions, etc. So let's talk about some of these things just to make sure that everybody's on the same page. Okay, who can tell me what this is? You can write your answers in chat. Sequence alignment, NGS, great. Hit map, multiple alignment, sequencing. Okay, all right. So some good answers right here, right? So what are we seeing? We are seeing here the output from next generation sequencing. And it is indeed alignment, but it's not multiple sequence alignment. This is alignment of reads reference genome. So these small pieces that you see here are reads. Those reads are short portions, anywhere between 30 to 300 nucleotides long, and those have to be assembled into a genome. So one easy way to assemble a genome is to map it on a genome that we know is closely related. So when we look at this, we see here a lot of short sequences. Those sequences are aligned to some kind of a reference genome. And then we have a consensus, which is the agreement, what all the reads mostly agree on in terms of a sequence, and we get a new novel beta coronavirus genome. So that's how we got from detecting some unknown reads from a host, let's say a bat, sequencing it, and then mapping it onto a database of potential viruses, and then understanding that perhaps there's a novel virus that is present here. So that is one aspect that we will talk about in the future. But right now, based on this process of next generation sequencing first, then finding a consensus of what all the reads agree to that a novel sequence is, we can then characterize a novel genome. And the novel genome looks something like that. So again, we have about 30,000 base pair long sequence. That 30,000 base pair long sequence is organized in a number of elements. And these are structures, so these are genes. Some of them code for proteins that are going to be important for the process of this virus replicating itself and entering into the cell. So here you can see what the sequences look like, right? So you can see that these are actually FASTA files, right? The previous reads that we saw were called FASTQ files, typically as an output from a next generation sequencer. But then what we are looking at is the FASTA files. And the FASTA files really just contain the nucleotide sequence. So what are we expecting to find in this sequence? Some of the things that we're expecting to find as a change from that reference genome are point mutations. So point mutations could be of several different kinds. They could be substitutions, where one letter is substituted by another. We can find an insertion, where one letter is added inside a sequence. And then we can see deletion. Now, these different changes in that nucleotide sequence result in different products. And those products over here are the different amino acids that are coded by codons. So the process works like this. Typically, there's a gene in a DNA. In our case, it's an RNA virus, but typically in the DNA, let's say in a human DNA, we have a gene. That gene sequence is transcribed to mRNA, which is messenger RNA, which is then translated into protein. And we kind of figured out the way this happens. It happens by taking these three nucleotide sequences, which are called codons, and we know which variations of those codons correspond to which amino acids, which actually form the protein. So you can see here a chart of how different positions that are changed have different effect on the product, on the protein that we have. Now, what are these amino acids? Amino acids are actually of different types. They have different chemical properties. So for example, they could be hydrophobic or hydrophilic. They can be charged or uncharged. Some of them have no polar charge. So these are different, types of amino acids are actually important for the structure of the protein. Let's think about it for a second. Here's the spike glycoprotein from this novel coronavirus. It has a certain structure. The structure is actually a structure of amino acids. So if we have polar charged amino acids, these amino acids have to support the function of this protein. They're there for a reason. So if they have to be there for a reason, what could be an important property, chemical property, that these proteins might have? Well, polar charge amino acids are hydrophilic and they have a charge. And they typically function in binding and catalysis. So that means that they do something. Why could this protein have to have a specific charge? The reason is because it actually connects. So you can see that this protein connects to a receptor. And that connection has to happen by using the properties of those amino acids. On the flip side, some of the uncharged amino acids might be facing towards the inside of the protein. So we will see how these different amino acids actually work to support the function of the protein that we are studying. For example, the protein that we started talking about, this spike glycoprotein, what it does is it actually helps in the entry of the protein of the whole virus into the human cell. And so we have different portions of each protein. So remember, we talked about the sequence at the nucleotide level. It had different sequences of genes. Those genes, in fact, have portions, and those portions are used together to form what's called cell fusion. So the two membranes of the virus and of the cell fuse together. And what is the point of that for the virus? Of course, it's the entry of the RNA inside the human cell. And the entry of that RNA is what causes disease, because that RNA starts entering a process that the cell designed for its own mRNA and starts the process of replication. So it produces a lot of itself. We'll talk about that in a little bit. But there are also other amino acids, non-polar amino acids, which are actually used as building blocks. So think of bricks. You can have functional things in your house. You can have a door, you can have a window, you can have the doorbell, you can have your fireplace, and those are functional things. But you also have to have walls, and those walls support everything. They make everything kind of stand together. And so those are going to be, a lot of those are going to be non-polar amino acids. Now, how do we go from the sequence that we detect by sequencing, right? We talked about sequencing short reads and annotating them together by putting them alongside some known sequence. But how do we know what are these different sequences and what do they actually do? Well, a lot of it has to do with protein function. Protein function can have multiple different aspects that we can study. One is it has an amino acid sequence. The amino acid sequence can reveal sequence similarity. For example, it could have a very closely associated known sequence that we already know what it does. It can have genomic context. For example, what typical portions it has. Is it in the beginning? Is it at the end? Does it have this feature or that feature? And it has feature-based. So we can have, for example, where typically it's found in subcellular location or a functional class. So these are some of the tools that we have to analyze amino acid sequences based on just the sequence of amino acids. Then we can look at the 3D structure. And the 3D structure, as you can see, is how these amino acids, if they are left in their natural environment, typically fold because they are charged, because they have certain chemical characteristics, they're going to form a certain structure. And so we can look at, again, different motifs and different characteristics of those amino acids to understand how they might fold, and that might give us insight into their function. Another way to study this is to look at high throughput data. So to conduct experiments by changing different things, and we can start detecting how different proteins interact between each other. For example, which protein typically binds to which protein. Or we can look at gene expression. So an easy way to do this is to sequence everything that we observe in a cell that, for example, has been infected, and then start looking at what does it do? What are some of the functional things that start happening to the cell once we start that process? So to do this, we actually have prepared multiple projects that have publicly available data so that you can study any of these processes. So for example, one of the projects that we'll look at in future webinars like this is the the identification of viral genomes in patient samples using next generation sequencing and comparative phylogenetic analysis. So we'll look at how can we take a sample from a bat or from a human and map all of the reads on, for example, the human genome or the bat genome to get a remainder of non-mapped reads that then could identify specific viral strains by mapping those onto a database. And then once we have those, we can do comparative analysis that will help us understand what they might be closer or farther away from. Another project that we'll talk about is the spike glycoprotein. We'll start talking a little bit about it today, but we'll continue talking about it in greater detail because it is the key to host cell binding, cell entry, and evasion from immune response. And we'll talk about how some of those are also the result of a very smart strategy by the virus in this case to avoid detection by the immune system and use different components of the protein. You can see that it has many components to adapt to the different challenges that it faces, to become more efficient at what it does, and ultimately to replicate in a way that it is undetected until it's already replicating a lot. And finally, we'll talk about a project where they took a specific cell type, the epithelial cells, that could help understand what kinds of proteins are being produced inside the cell as a response to different challenges by these types of viruses. So what they did is they took one of the genomes and modified one of the genes in that genome, changed something in the viral genome, infected the epithelial cells, and started comparing, well, what happens between the mutant and the wild type, and how can we understand that change? And so what you can see here, and we'll talk a lot about this in future sessions, is a principal component analysis that helps us characterize that there is a difference. And there is a difference here between the mutant and the wild type, and at different time points. That's another thing that we have here. So a lot of great data is available online, and we can use very effective tools to study this type of process at a very detailed molecular level, which is, I think, very exciting. Okay, so before we go and we start finding the genomes of other coronaviruses, I want to pause here and ask if anyone has any questions about what we covered up until now. All right, great. So if there's no questions, let's keep on going forward with finding that data and seeing what we can do with the data and not just learn about it. Okay, so let's remember some of these key things that we have multiple strains of the same beta coronavirus. They will be called beta coronavirus 1. They could be called MERS, SARS, or the novel coronavirus. Let's remember that the coronavirus genome is going to be approximately 30,000 nucleotides long, and it will have specific regions that are going to be important to understand how this virus works. So when we talk about comparing those sequences, we can compare whole genomes between each other, but that's a lot of data to compare, right? That's 30,000 nucleotides. Or we can start looking at very specific positions that we think might be more relevant to how it is different. Okay, so let's first talk about multiple sequence alignment. What is that? Here's a multiple sequence alignment. Please take a look at this and tell me, what do you think we are aligning and what are the differences that could be seen? Please answer your, please put your answers in the chat. Okay, great. Great. DNA sequences. Yes, so this is not amino acids anymore, right? This is right now, exactly, different human versions of which coronaviruses. What do we see here? Yes, different human infections, DNA sequences. Okay, so let's break this down. We have here, right, so we have a bunch of different viral sequences. We can see that all of them contain this severe acute respiratory syndrome related coronavirus, right? And then we have where they came from, their location, USA, China, Spain, Israel, USA, etc. And we have the date, right? So we have the 2020, 2019, 2004. Okay, so different dates. And we have one that is used as a reference. So now let's take a look at this side of the picture. So what do we see here? Here we see on top is the quality, right? So this could be called the quality of the sequence, of the consensus sequence. This is the consensus sequence. Or we can also call it the frequency of mutations. So we'll talk about some of these things now. So we have a letter, for example, in all of them it's C, all of a sudden in a bunch of them it is T. Why do you think there is this change from C to T, from C to A? Why do we see these white boxes? For example, in 1, 2, 3, 4, 5, 6, 7, in these 7, what do you see is different in these 7 from the rest of them? Okay, mutations, but why? Why do you think that is? What is different between these and these in the middle? Change of country, well, there's USA right here, there's USA. What else is different? Age, we don't know the age. SNP, okay, what do we see here? What is this? Year, all right, great. So we have different years. So what do we know about the years? The novel coronavirus started in 2019. Remember, it's COVID-19, right? So in 2019, so 2019 to 20 are the novel coronavirus. And then these are the old SARS coronaviruses that are not the novel coronavirus. Okay, so we're starting to see that there are changes, consistent changes, right? Consistent changes between the older coronavirus and the novel coronavirus, or the SARS coronavirus. Okay, so that's what multiple sequence alignment actually tells us. But as you can see, this is a very small portion of the full genome. So some other ways that we can look at these differences, we can look at mutation frequency, right? Mutation frequency tells us how many samples contain a change based on the reference genome. So for example, here you can see that there are some changes. This was the segment that we were looking at in detail. Sometimes the detail is not interesting. We just want to quantify it. And so we can see that 8 out of all the sequences contained this change. And that might give us an insight. We can also look at, okay, well, if there's a change in the nucleotide, does it actually cause the change in the amino acid, right? Is this a mutation that causes change in the protein that is being produced? If it does, maybe that's more significant than the change that we observe at the nucleotide level, right? So that's where we have the amino acid mutation frequency. And it looks something like this if you zoom in. But you can also map those mutations, those differences, on a 3D structure. So here you can see that spike glycoprotein from the top, and you can see this section that is enriched with mutations. So mutation frequency, you can see it is enriched with mutations. Okay, what kind of graph or map is this called? So again, this is multiple sequence alignment. This is the quality of consensus sequence. This is based on clustral multiple sequence alignment. And here you have mutation frequency at the nucleotide level. This is mutation frequency at the amino acid level. And this is the transposing of that onto a 3D structure from the PDB file 6VXX. And that also shows mutation frequency. Okay, we'll talk about all of these things in greater detail in some of our future sessions.
Last year.
Okay, here's another concept, a phylogenetic tree, right? So we can detect these different mutations, we can point them, we can annotate them, characterize them. But first, before we go into such detail, we want to understand, well, how does it in general describe the data that we have? So here we have a phylogenetic tree. And what does the phylogenetic tree tell us? The phylogenetic tree tells us how we think the present day species that we observe, right? So for example, there, we were looking at the, we were looking at two types, right? The SARS from the previous years and SARS from 2019, 2020. So that's the present day species that we observe. But our question is, where could they have come from, right? So they have branches that come from a source, and we think that that was the origin, right? So if we want to measure distance from origin, and we think that they might have come from a common ancestor, we link them together. So that's what a phylogenetic tree tells us, and it helps us organize our information in a certain taxonomy. These same phylogenetic trees could be rectangular, circular, radial, so they can have multiple versions, but they all speak about the same thing. They talk about the relationship, and they might include some kind of a distance, right? So let's take a look at an analysis of a bunch of these different coronaviruses from that relationship perspective. So here we see the human coronavirus HKU1, which is a beta coronavirus. You can see that it was discovered in 2005 in 2 patients in Hong Kong. So they're right here. We have the beta coronavirus 1, which is, which again is very closely related, right? You can see that it comes from a common source. Then we have MERS. MERS is hypothesized to have come from camels, maybe also bats, but you can see that it was first reported in Saudi Arabia in 2012. It had severe respiratory illness and a lot of deaths. So actually high probability of dying from that disease, and it's its own clade right here. You can see its own clade. Then we have the SARS coronavirus, which was first reported in Asia, again in China in 2003. It was contained, and no novel infections were detected after 2004. But you can see it also forms its own little branch right here. And we have the novel coronavirus, which is very closely related to SARS, right? So right here, close in the same clade. We can link this information, like we were looking at, to where this came from. So a very nice website that we can all learn from is called Nextstrain. So let's quickly just take a look at that link so that we can understand how these things are related. So you can go to Nextstrain. And it has several different viruses that you can look at, but let's take a look at it now. And here, sorry, that was to everyone. Okay, so please take a look at that link. Here you can see how starting from 2019, this virus has evolved, right? So accumulation of mutations in these genomes, their evolution, and you can play here how it occurred and link the data at the genomic level, right, to the spread that happened over time. So here you can see that it started spreading from China. More samples were collected in China. Eventually, samples were collected in Australia and Japan. Then it went to America and to Europe. So you can see that the spread happened, and we have all of these different sequences from these different parts, right? And now that you understand multiple sequence alignment, you start understanding this chart right here below. So you can start seeing emerging mutations, right? So these emerging mutations down below characterize these different substrains of this novel coronavirus. You have some options here on the site that I encourage everybody to try and play with. You can see here the different data sets, You can change your settings and kind of see how it relates to some of the other things as well. So fascinating way to connect all the data together, but the key to this connection is really the analysis. So we don't have that much time left, but what I want to leave you with is to start asking that question. Another place that we should go to is a place where all of these sequences could be easily found, and you can start analyzing them. So this is the link that we want to go to. It's the the NCBI virus. You can click right here, search by virus, and type in coronavirus. And we want to look at the beta coronavirus together. So this is the link to get to the beta coronavirus. I'm going to paste the link right here. And let's take a look at some of this information that anyone, including yourself and me, we can find and we can start looking at. So here you can see the accession numbers. You can see the length of their genome, the geolocation. You can see host isolation, where they were isolated from, and the collection date. So, oops. Okay. So there's a lot of data here, right? Obviously, you need to filter this data. So for those of you that have a little bit more time, I'm going to walk you through on how to select a meaningful data set from here and show you how to do a very quick, simple analysis to understand this relationship that we saw from phylogenetic standpoint, right? So the first thing that we'll look at is phylogeny of these different sequences. Okay, so what kind of information do we have? We can download this information and start filtering it in different ways. We have the strain, country, host, the GenBank accession, and we can start grouping those by a specific variation that we can identify. So let's think of how to do this quickly. To do this, I'm going to demonstrate it to you here. If you're going to be joining future sessions, we're actually happy to create an account for you to try and play around with some of this stuff. What we'll do is we will go to our platform and we'll try to build the phylogenetic tree with one of the examples that we prepared for this session. So the platform link is server.t-bio. The benefit of this platform is that it allows us to do fairly complex analyses on a variety of different data types. Many of these same algorithms are implemented in different ways on different solutions. So I'm not saying that this is the only way to do it. In fact, you can build a phylogenetic tree right there on the Virus NCBI resource. But here, the benefit is that this is going to be a resource linked to your students' projects, and you can monitor all of their activities. If you are a teacher in a school, you can monitor all of their activities and link them to specific projects and courses that we have available online. So in this whole section, you can see that we have next generation sequencing data, we have mass spec data, structural biology data, and then data integration and modeling. And so we're not going to be processing right now the raw reads from sequencing. What we will do is take prepared genomes, and I'll just show you a quick demo to leave you with some idea of how you would go about it yourself. So we will go here in the analysis of virus quasi-species, where we will do multiple sequence alignment and phylogenetic analysis. So we will start by taking a list of sequences from that virus database that we just looked at. Remember, the paper that we're following took 3 different bat genomes, 1 pangolin genome, and a couple of human genomes. Then we will go to multiple sequence alignment at the nucleotide level. After that, we will convert that alignment into amino acid alignment. So we want to understand which nucleotide changes cause the change in the amino acid. And we will annotate those by their codon position in the different genes coding for different proteins. Then we have here implemented a number of different phylogeny methods, but what we will use is beast and ape, and we'll talk about those methods more later, but we'll build a phylogenetic tree. And then we'll analyze those phylogenetic trees to select the best one that we think is representative of what really happened. Okay, so now we can take a look at what some of those outputs look like. Here you can see a number of different outputs. Some of them are FASTA format. So you saw the multiple sequence alignment was in FASTA format. Some of them are going to be tables that you can open in Excel, for example, and look at frequency of mutations and things like that. Some of them are PDFs that show us the different outputs in a phylogenetic tree visualization. So let's take a look at this PDF. We'll just download it like this. And let's see the result. So here, tell me how many distinct groups do you see in this visualization? Four. Anyone else? Four. Great. So we see 4 clades. 4 clades, and the question is, what are they? Right? So how do we know what they are? How could I find out what, for example, is common between these genomes or common between these genomes? Great. Thank you, everyone, for your answers. How do you think I could actually have a sense of what these groups are? Okay, analyze, we already analyzed, yeah, accession ID, exactly. So these codes are accession IDs. So let's try and find them real quick to understand what this means. So where is our virus, NCBI virus? Okay, so this is the accession ID, right? Let's just search for it like this, and we'll find it on NCBI. Let's go to NCBI. Okay, look for this. Okay, so what this is the human coronavirus OC43 from USA from the 1990s, complete genome, right? Okay, so let's first look at the accession number, right? And then we'll look at why. So all the other answers that you guys had is why, right? So we can analyze our sequences to understand how are they different. We can annotate the different proteins to understand the functional differences, etc., right? But at the first level, we just want to understand, okay, did this produce some kind of a meaningful result? Okay, so we noticed that the first one is the beta coronaviruses from the 1990s. Let's take a look at this one. Because I had a space. Oh, that's a K, right? LC. Okay, so here again, we will have the nucleotide, sorry. Where was it? Nucleotide. All right. Let's Okay, this record was removed, but you can see that this was novel coronavirus 2 from Japan. And so forth, right? And we can see outgroup frame stands for the reference genome that we used. And as you remember, this is the reference genome that we originally used for this novel coronavirus. So to simplify the story, let me point you to another resource that we developed that can help link all of this information together. So here you can see how the frequency of mutations, right? So here we see, we can play around with some of the tools that we have here. Let me just send this to everyone. Okay, we see here the protein sequence and the different proteins that you can look at their 3D structure. You can learn about what each protein does. And you can, of course, look at what kinds of changes occur in that protein sequence based on the nucleotide sequence that we see right here. So for example, here we see the reference genome. The reference genome is the novel coronavirus. And here we have different strains. You can click on each one and see where it's from. We have here different beta coronaviruses. So you can see that COVID-19, SARS, beta coronaviruses, and MERS. And there's a couple of different examples right here that you can switch between. So for example, how do all the COVID-19 strains compare, or how do different types of coronaviruses compare? And you can see that the frequency of mutation is going to be marked by either gray, which is 0, or red, which is a lot. And that will vary between different bins. And by looking at this overview, you can really see how you can start seeing the significance
Hello?
of change in more
Hello?
general terms between
Hello?
what we have in general, right? The distance between these different viral genomes. So this is where I think we can go further in our future sessions. And the next session that we plan to organize is going to be related to the different proteins that we plan to analyze to better understand how the virus enters the cell, how it replicates, and what would be the different outputs from these different algorithms that help us analyze this data. So I just wanted to get this presentation to a stopping point before we open this for questions. Now I see that a lot of you have questions, so let me pause here and start answering some of those. So first of all, Hermaine Raiz was asking, if you want Genome, then can you download it from Ensemble, and what do you want to do? Finding mutations between different strains of coronavirus. Okay, so great question. What we plan to do, right, is follow a paper that we started talking about, which was analysis of amino acid mutations in specific protein sequences that can help us understand how different sources, potential sources, and potential hosts where the virus came from would give us an insight into the process of transfer of this virus from different hosts to the human. Sorry. Okay, the second question, can I share the presentation? Yes, I will be happy to share the presentation. We will email it to participants that are interested in this with a survey to better understand how this might have been relevant or useful for what you do, because we do want to understand how to further advance these types of resources and adapt them to the need out there. We also want to understand how next sessions could be useful for you and your students. So as I mentioned, in our next session, let me give you a little overview of what we will cover. In our next session, we will talk about the biology of coronaviruses, where the replication is facilitated by different proteins. And by analyzing the different proteins, we will better understand how the changes between different viruses result in, for example, higher infection rate, evasion from immune response, and others. And we've prepared a number of different resources that we didn't have a chance to talk about today that really go deeper into the structure of different proteins, the different functional domains of proteins, and we'll in detail kind of understand how does this virus actually work, what does it do with these different proteins, and how these mutations can help us answer some of the questions that we might have about the viral process. Okay, some of the questions that are posed here. Why don't we use BLAST? Yes, for sure, you can use BLAST. BLAST is local alignment, so it's not going to work well for the full genome. So for full genome, you use multiple sequence alignment. What is TaxID? TaxID, as we looked at, there's a taxonomy of these viruses, so we use that to associate them with different substrains, genera, species, etc. If a virus still shows mutation, how would one design a vaccine? Yes, that's exactly where we're going with all of this. We'll talk about knowing the position of the mutation will give us a lot of insights into vaccine design, but we first have to go through the steps to get there. By this analysis, can we identify some hotspots that are more prone to mutations studying different strains from these outbreaks? Exactly, that's a very good point. In fact, we will talk about the receptor binding domain that will show differences between different hosts and different strains of the virus, and an important part in vaccine design. Can we develop an inhibitor? That's a very good question. And a good question is to ask yourself, inhibitor of what, right? So an inhibitor means that it slows something down. What do we want to slow down? We can slow down virus entry into the cell. We can slow down replication. If we have replication, it is not done by the same protein, right? So there's another protein that is responsible for different aspects of this viral replication. So we need to look at the different proteins, ask ourselves, what kind of data set can answer a question that you might have? Okay, what cell types express ACE2? Epithelial cells, just off the top of my head. You can definitely look into it. It's a good question, yes? So we will talk about next time how this protein, the surface spike glycoprotein, connects to the ACE receptor, and we'll talk about that receptor in detail. All right, great. So good questions, everyone. Thank you again for joining. Let me just finish with one more slide. We do encourage all of you to think about these questions that we started looking into today. We showed you today some of the tools that you can use. For participating in science courses next school year, I would recommend connecting with ScienceCoach. So ScienceCoach is a program. You can see the chat message from Jill. There's a specific website called sciencecoach.org where you can learn about ScienceCoach. If you're interested in bioinformatics, then you can go to sciencecoach.t-bio.info. And we have a lot of different questions answered, thank you, Beepsha, on our blog post, where a lot of these questions are addressed in detail. Yes, Dipanshu, very good idea. I think this is something that a lot of people are looking into. All right. And we will definitely send you more information and the recording of this webinar. So this is what we have in mind for our next couple of sessions. If you would bring your students, your kids, or join yourselves to learn more about ScienceCoach and bioinformatics for COVID-19, we would be happy to have you again. Thank you again, and have a great day, everyone.
Bye.
Yes. Sorry, last question. Is it paid or is it free? So ScienceCoach has a program for teacher training. We will probably send you more details by email about teacher training available from ScienceCoach. Thank you.